जय हिंद एवरीवन। मैं पंकज कुमार और आज की क्लास में हम लोग देखने वाले हैं। क्या देखने वाले हैं आज की क्लास में? आज की क्लास में हम लोग देखने वाले हैं एक्सक्रीटरी प्रोडक्ट एंड देयर एलिमिनेशन। So this class will be in English only. Don't worry about the language. We will wait for 1 minute, and if anybody of your classmates, friends, relatives, anyone want to learn how that excretory product will be eliminated from the body, you can share this link to them. Okay. The class is started now. Also, screen is visible. What is this happening? Okay, screen is visible. Now, shall we start? Shall we start? Shall we start? Shall we start? So, in last class, what we studied? We studied about the excretory product, excretory organ in the different organism, and what are the types of the excretory. Uh, what are the wastes? There are so many type of wastes, but majorly the organism are divided according to their waste product. Like if they are excreting the waste, like ammonia, then it is called as ammonotelic, urea, then ureotelic, and uric acid, then uricotelic. So these are the important uh, type of the organism. Okay. So let's see the urine formation in this cluster. Kidney and urine formation we will see. So without delay, we'll draw one kidney and we'll see the structure of that. This is your okay. So you can see this beautiful diagram as I drawn. So these are the arteries and these are the veins. Why we need a vein? Because the kidney cell itself it required oxygen to perform a function. So that during the respiration, this kidney cell will produce the CO2 and that will be transmitted by this veins. And why we are taking this arteries? Again, two reasons. The cell itself it will perform a function, so it required oxygen. It will supply the oxygen as well as the important point. It will filter the blood also. What it will do? It will filter this blood. It will do the filtration of this blood. Got it? So the region where it is making a notch-like structure, this region you can see, this region which is making a notch-like structure, that is called as a hilum. What it is called? This is called as a hilum. From where these arteries and veins will be enter inside the kidney. from where these arteries and veins will be entering inside the kidney. If you see, these arteries and veins are entering, and these arteries will be, it will break into so many arterioles, and finally it will go inside this, from this wide region, from from here. And this wide region, this is green color region, is called as a, this will called as a pelvis. And from this wide region, it will go to the major calyxes. Okay, it will go to the major calyxes. I will draw this. The region which you are seeing with the yellow color, that is the cortex. Okay, that is the cortex of the kidney, and the region which is completely white, that is the medulla. And the triangular shape which you are seeing, that is a, what it is? Medullary pyramid. So this is the very important topic, because this have one important concept, that a RAS, renin angiotensin pathway. Okay, and it will related with the so many different organ, like adrenal gland, then vasopressin, like posterior pituitary gland, and the regulation of the blood pressure is also here only. Okay, so that is a very important concept. They can ask the questions from it directly. Now, the region you are seeing here, this region is called as a column of Bertini. This gap which you are seeing, this blue color gap which you are seeing, this is called as a renal column, you can say, or column of Bertini. Of column of Bertini, and this is a medullary pyramids. This is the medullary pyramid, and the region which you are seeing, this I should make with a different color. I will take this. So this region either it can be wider or it can be little narrower. So this region with blue color, it will called as a calycis. What it is called? Calycis. Or it can be wider, then it will called as a major calycis. Or if it is a narrower, then it will called as a narrower, no, minor calycis. Okay. So these regions will called as a, so this region will called as a calyx or major or minor calycis. Okay. So this is the important thing which we are going to, which we are seeing now. And now, if you see anything else, arteries you see, these are the arteries, these are the veins, arterioles and veins, and then it will going into the hilum, this hilum region, and from hilum, this green region will called as a pelvis. Pelvis. From pelvis, the renal tubule, you can see, so these arteries will be divided into so many capillaries, and that small, sorry, tubules, and then it will pass over the calyces, major calyces, and then it will enter to the column of Bertini. From there, it will go to the renal, and there it will go to the yellow region, which is called as a cortex. And from this cortex, the renal, that pyramid, you can say, renal pyramids, which are present in the space of medulla also, or and in space of the that boundary. This boundary is the space of this yellow boundary also, and this space of the white boundary also. So that is a renal boundary. The yellow boundary is called as a cortex, and the white region, which is, you can see, the middle region is called as a medulla. Apart from that, you have to find the important things. What are the important things about this kidney? The structure will be of around 10 centimeter long, then and then width will be 5 to 7 centimeter width and 2 to 3 centimeter thickness. 2 to 3 centimeter thickness, and it is divided into two parts: the cortex and medulla. Its weight will be around, together it will, the weight will be around 120 gram to 150 grams. If you write, okay. That is the range. So you have to write the weight of this kidney. So what do you think? What will be the purpose of designing this kidney? Why God designed this kidney? What will be the functions of the kidney? So you can text in your chat box also, or you can share it directly. Okay. So the functions, we'll see the functions. The function of the kidney. The function of the kidney is first is what? The first, it will help in the osmolarity. Okay. Osmolarity. It will balance our osmolarity, maintaining the blood pH. So if I am saying osmolarity, it will consider all the electrolytes and the water content in our body. It will be maintained by the kidney. Okay, how much salt is required, how much water is required in the body, and that adjustment will be done by this kidney only. Okay. Along with that, as you know, the plasma of the blood will decide the pH, and in plasma only water content will be there, and then water content will be controlled by this kidney as a in terms of the osmolarity. So along with that, it will affect our blood pH also. So it will maintain our blood pH. Third thing, what is the third thing? It will regulate our blood pressure also. Okay, it will regulate our blood pressure, and the important thing, if you this is very uh, it is very unnatural to listen that kidney will be decide how much glucose will be you have to be breakdown, or how much glycogen have to be breakdown. So in simple way, it will decide the level of the glucose in the body also. Okay, so it will help in the maintaining glucose level also. How? It's not direct but indirectly. Okay, it's not direct but indirectly. What you can write? Regulate glucose level. Okay, regulate glucose level. If you think how the kidney will be used for the kidney will be used for the synthesizing of the red blood cell, and that red blood cell is synthesized usually where? In the bone marrow. So there is some important hormone like erythropoietin. Okay, erythropoietin, which will be released by the kidney only, and it will helps in the RBC synthesis. It will help with the RBC synthesis. One more hormone it will release, that is a calcitriol. Okay, it is calcitriol. Actually, calcitriol is a active vitamin D. Okay, what it is? It is active vitamin D. So these are the important things which will be done by the kidney, and filtration. the important thing which we are going to read this topic, that is the filtration of the blood, and reabsorption, secretion, removing excretion of the waste. Okay, excretion of the, oh shit, something happened wrong. Excretion of waste. The waste, if I am writing the which kind of waste it will, it can be either protein waste. Okay, so like protein waste will be the ammonia and all. It can be nucleic acid waste. Okay, acid waste. Okay, or it can be either creatinine, or either it can be urea. Okay, so there will be different type of waste which have to be removed. If you will think that H plus is also waste, it's not actually waste. This H plus reabsorption and absorptions will be reabsorptions and the secretion will be occurred because of the maintenance of the pH and just because of the maintaining the osmolarity. Okay, so this is the important things, the important functions of the kidney which you have to remember always, and it can be asked the important points if you want to highlight that erythropoietin. They can ask the role of as a pH balance. They can ask. Okay, and this is the important thing which we will read in this class. The how it will regulate the blood pressure. Okay, so for today's class, this much only. We'll continue the our class. Your response, if you are liking these classes, you must just show your appearance that yes, you're liking and you're watching these classes. So till now, if you have any doubt, you can ask me. We are not going to finish this class. Wait. Okay, till now, anybody have any doubt? Shall we continue the class or shall we stop here only? Because uh, this is the one side, like I'm not telling you the monkey bath. Okay, what I did, what the policies we made, what I did at my home, what is happening. It's not my monkey bath. It's like the two ways of conversation. You should reply in your chat box also. You have a opportunity. You can ask your doubt till now if you have any doubt. If you don't have any doubt, just give this like buttons. So I will be understand. You don't have any doubts. Where are the emojis? Don't know. Wait. If you have any doubt, you can ask. If you don't have any doubt, then you give just like buttons. There was one thumbs up. I'm unable to see here. Okay. Ah, smiley you can give. Yes, if you don't have any doubt, you just send it. Then only I will understand. Now. Oh, shall we start? Shall we continue this class? Are you feeling boring or like something? No. Give the chat. I would like to see chat. Shall we continue the class? Chat is open or not? Okay then. So we'll continue this class. So now the thing is that we have to read the structure. What we have to read? We have to read this structure of the nephron. Hope so you understood till now the function. Again, you remember it, the function like erythropoietin, it will release calcitrol it will release, and in coming soon we will see how it will maintain the blood pressure. So structure of nephron. So for past 10 to 20, like 10 years, they didn't ask this type of questions. Okay, what is the structure of the kidney? What is the length or the wideness of this? They will ask the question from the structure of the nephron or the blood urine formation. So this is the suppose I am going to make a renal pyramid. Okay, this is my renal pyramid, and here wait, leave this renal pyramid. Simply, this is your Bowman's capsule. Okay, and this Bowman's capsule is the part of nephron, and it will continue with the tubule that is called PCT. And if you see this continuation of this tubule, this is very important. So for reabsorption, and now we'll see this is the widened part, and this widened part will become completely narrower if it will go down. This is called as a Henle's loop of Henle, and later it comes up. This is GCT, and then it will go to the collecting bed. See, now I will tell you most important topic in this only. So don't go and leave this class. Okay. So this is the typical nephron which you already studied in your 9th, 10th class, and this is the afferent arteriole. Suppose this is the afferent arteriole, it will be divided into so many capillaries, and these capillaries will be very narrower to increase the blood pressures. So these are the capillaries. And I will make it like a blue, so you will understand. Tubule. This is the same tubule, but it is different. Okay. So blood will come from the this side, left side, and from where the blood is coming, that is called as a afferent. And where blood is going out, that is called exit. And exit starts from the E, and exit will tell you the efferent. Okay. So efferent, remember it, it is exit, and afferent where it is coming inside. So this whole diagram, this complete diagram altogether, it will be called as a Malpighian body. What it is called? Malpighian, Malpighian body, and this is called as a Bowman's capsule. Bowman's capsule. Okay, and this is called as a glomerulus. This will called as a glomerulus. Yes, glomerulus, and Malpighian body part is done. So another part is the renal tubule. So you can divide this nephron in two parts. That Malpighian body, which is consist of the glomerulus and the Bowman's capsule, where the filtration will be occur, and the remaining is the renal tubule. The renal tubule is divided into the PCT, proximal convoluted tubule. DCT means distal, which is backside. Proximal, which is in the front. First, what you are seeing, that is a PCT. Proximal convoluted means what? It is just like you can say ups and down. This it's not straight going ups and down like that. It is a convoluted. Okay, and distal DCT distal convoluted table. It is in the back side, distal side. Okay, that is telling the position only. Okay, so it is this PCT and DCT is connected with the loop of Henle. What it is? It is connected with the loop of Henle. loop of okay. So hope so you understood till now. And last, you can see this is a collecting duct. Collecting duct. Okay. You just see this diagram. So here the arteries is coming, and arteries will be break down into small tubules. Okay. So this arterioles will be goes into the column of Botany, and then it will supply this to the nephron. And where nephron will be present? Nephron will be present here. I will draw with a green color, and this is the nephron. Okay. So nephron is present here. This is like this, and most of the nephron. Okay, this is the nephron. Most of the nephron will be in the cortical region, in the yellow region. Okay, very rarely, like not really, but some of the nephron which will be goes like this, and its renal loop of Henle will go very deep into the medulla. So it is going very deep down. Okay, so if you will see this nephron is called as a juxtamedullary nephron. Okay, what it is? Medullary nephron, you can say directly, and cortical nephron. So this is your medullary nephron, and it will be only 20%. Around 20 to 25%, we cannot give the exact value. That is a range. And this will be what? This will be your cortical nephron. Okay, cortical nephron. And this cortical nephron percentage will be the remaining will be how much? 80% only. So 80 to 85% will be the cortical nephron, and medullary nephron will be 20%. So what is the main meaning of the juxtamedullary nephron and the cortical nephron? Medullary nephron or juxtamedullary nephron, if I am saying this is both same. So this depends upon this loop of Henle, how much deep it is going inside. Okay, how much deep it is going inside. If it is going till the medulla, then it is called juxtamedullary, and if it is in the most of the part in the cortex, then it is called as a cortical nephron. Always, this PCT and DCT and this Malpighian body will be in the cortex side. Okay, this will be in the cortex side, and loop of Henle collecting duct will be in the medulla side. Okay, so always loop of Henle length will be short in cortical nephron. Now finally, whatever the blood is coming, blood will be filtered here, and filtration depends upon the pressure of the blood. Okay, so after filtration, Bowman's capsule will take over the waste, not only waste, all the filtered part. So the filtration may contain glucose also, may contain amino acid also, may contain any drug also. So it have to be reabsorbed. So the two things will be happen here. If I am saying reabsorbs means the fluid which is passing in this, that have to be the fluid which is passing in this, you have to extract from this. Okay. So this is the fluid. If I am saying reabsorption means something you will take from this. Okay, this is arrow will tell you the reabsorption. Okay, reabsorption. This is a very important point to understand what is reabsorption and what is secretion. So if I am saying here, in case of this urine formation in this chapter, if I am saying secretion means you are adding something into this filtration. You are adding something in this filtration, and where should I write secretion? Wait, don't have a space. So I will write here. Okay. If I am saying secretion, it means you are adding something in this. Okay, this is the secretion. Okay, if you are adding something inside this, then it is called a secretion. If you are taking out of this, then it is called as a reabsorption. Okay, so it is same like that. If you are washing something in your kitchen, what something suppose vegetables, cut vegetable. Okay. If something important like vegetable will be two, three vegetables, cut piece of the vegetable will fall into the basin. What you will take? You will take pick that vegetable piece from the basin and you will wash and you will keep into the your utensil. Okay. But if there will be something in that utensil, that waste, like leaf or any insect or anything. So what you will do, you will pick that insect or leaf and you will throw into the basin. It is simple only. Reabsorption, what you did, you vegetable piece, it by chance it will goes into the waste like in a renal tubule. So it will go to the collecting duct and you will pass out from the urine. So you don't want to pass out the glucose, which is very important. So what you will do, you will pick that glucose from the basin, means from the tubule. That is called reabsorption. And if by chance more H plus or potassium will be there in your surrounding, then you have to secrete, means you have to throw out throw out that potassium H plus and some salts inside this tubule. And this will help in the maintaining the osmolarity. Hope so you understood. Any doubt is there till now? Any doubt? No doubts? Do you have any doubt? No? You understood? Okay. This thing we can see. Why am I? Okay. No doubts are there. Then we'll continue. Okay. So this you understood. Now the important thing we are going to see, that is a called as a mm, that juxtamedullary apparatus. Okay. That is it. Juxtamedullary apparatus. Ah. Okay. Medullary nephron, cortical nephron, and now we have to see why it is not working. Wait. Okay, come. JG juxtaglomerular apparatus. Okay. Apparatus. This is important thing, and this is structure you have to understand. This is the typical nephron, but this nephron will be not looks like this. The original nephron will looks like this. Okay. Now you see the original nephron how it looks. So this is your Bowman's capsule. Okay, I will make little small diagram. This is your Bowman's capsule. Small means small, not very large. This is Bowman's capsule. This is what it is. Tell me name. This is PCT. Then loop of Henle. Okay, and this is DCT. Now the beautiful thing is that instead of going like this, we will go opposite. Okay, how will go? We go like this. I will go like this. This is PCT. This is this is loop of Henle. Okay, this is loop of Henle, and now this is our DCT. Okay, did you seen this type of nephron? No, no. This is the first time you are seeing, and this is your afferent arteriole. and this is your efferent. Okay. So this is called as a juxtaglomerular apparatus. Okay. What it is called? This is efferent. I am blue color. I am making with this. That blue color line is the efferent arteriole. Okay. So the region what you're seeing here, the shaded portion. Okay. This is called as a juxtaglomerular apparatus. Okay. And this is called as a JG apparatus. So if you'll make it little broad, so what you'll find there is one DCT. Okay, there is one DCT, and the the surrounding this surrounding this arteriole is there. Okay, which arteriole is there? This arteriole is there. Okay, and they are in contact with the special type of cells that is called macula densa. Okay, so in arteriole, you will find the special type of cell that is, I'm going to make it with the red cell, and that is called as JG cell. These are the JG cells, and these JG cells will be only going to release that erythropoietins and renin. Okay, these are the JG cell. These cells will release the renin if if the GFR will be less. So how this command system will be go, that we will not read now, but we will see what are the cells in this. This is our DCT. What it is? Again, I will tell you. This is the DCT. So DCT have a special kind of cells that is called as macula densa. What it is called? It is in the DCT only. These are macula densa. Macula densa cell. Okay. So macula densa cell will like it will check the sodium concentration. What it will do? It will check the sodium concentration. Whenever the sodium concentration will decrease, it will give the signal to the JG cell, then it will give the signal to the renin, then it will like that it will go. So usually this will tell the it will check the sodium concentration, or we can say that GFR. Okay, if sodium concentration will decrease, then the GFR definitely it will be going to decrease. Then it have to be tell this all regulation that to the brain, so they will release some aldosterone, or sorry, brain, they will release the vasopressin, and then finally we have to increase the blood pressure, or we have to decrease the blood pressure. This will be accordingly it will be occur. Okay, so JJ apparatus, maybe you understood, juxtaglomerular apparatus, in which the three things will be there, and in between the three cells are the what, macula densa, JG cells, and in between that there is a one tissue will be there. Mm, there is one tissue. Okay, make it with this. In between this will be one tissue. Okay. So hope so you understood what is the important thing. These are the important thing you have to understand. JG apparatus you understood. After this, we will go for the reabsorption and the secretion. We'll see directly that, and what is GFR we'll see, and we'll see how that secretion will be occurred. So let me know. Anybody have any doubt? So you can type here. What you have to do? TYP type here. You can type here again. Anybody have any doubt? Shall we continue? Shall we continue? Cover this topic up to the urine formation. Anybody have any doubt? No. At least say no. No. 100%. No, sir. Shall we continue? No, sir. We should not continue. If you want me to continue, just give me this emoji. Where is that emoji? We have a lot of emoji, but okay, smiley. Good. Smiley we can go. So smiley we can go for it. Okay. Good. So we are going to continue this class. So up to here you understood JG apparatus. In coming classes, we will see what is the use of that JG apparatus. Okay. And don't draw nephron like this, which you are seeing here. Draw nephron like this. Okay. Actually, it is not correct. This is the actual nephron, and it's tough to explain DCT, PCT. That's why we are making a simple nephron diagram. Okay. So let's see what is the GFR. Okay. So what is this? What is this? Why we are reading here? Okay. What is this? GFR. So GFR is a glomerular filtration rate. Okay. Glomerular filtration rate. So if you see the glomerular filtration rate, then you will find there is a blood capillaries. Glomerular capillaries, and these are the cells. And okay, these cells have a small gap. Okay, these cells will have a small gap from which this big this these are the blood corpuscles. Okay, like RBC, WBC. This cannot pass from here. Okay, this cannot pass from here. But what can be passed? The small particles. Okay, which can be fit here, which can be fit here, which can be fit here. Okay, these are the small particles. And what are these small particles? The small particles can be like glucose, amino acid, urea, ions. Okay, these all can pass from here, and this is your endothelial membrane. Okay, what it was. Okay. That will be the yeah, it is. So these small particles can be passed because it have the size to fit into these gaps, and then it will be passed from here, and after passing it they will go through the basement membrane, and this is your basement membrane. Make it a little big. Okay, this is your basement membrane, and from here they have a epithelial tissue in the glomerulus. Okay, they have a epithelial tissue, and if you see this epithelial tissue, this This is the epithelial tissue, and that epithelial tissue is modified, and it is called as a podocytes. It will looks like this something. These are what? These are the podocytes. It will see these podocytes. This is the basement membrane. Basement membrane, and this is your endothelial tissue. Okay, endothelial endothelial layer. So podocyte, which is the epithelial layer, and that epithelial layer is modified as a podocyte. And why it is modified? Because it have to make a pore between this. Okay, what you have to make? You have to make a pore between this. So these are the pores from which these solute particles, this will be enter here, will be enter here, it will be enter here, and it will be enter here. So what should be the criteria to be entered here? First thing, it should be the small in size. Okay, small in size and fit into the gap. Okay, and fit into this pores. Okay, so this pore slits will be there. This will call as a pore slits. So it have to be fit into this pore slits. Once it will be fit into this pore slit, it will be pass over this uh, endothelial layer of the blood vessels, and then basement membrane, and then epithelial layer. Another thing, it have to be higher pressure. Okay, what it have to be? It have to be little higher pressure. That glomerular hydrostatic pressure we are saying, and osmotic pressure we are saying. Okay, it have to be. Shall we read? No. For you at your level, it's not required. Just we will write the arterial pressure should be higher. Okay, arterial pressure should be higher, which is called as hydrostatic pressure. Okay. Pressure. Yes, arterial pressure should be high. If this pressure will be high here, then only this flow will try to skip into this. Okay, pass into this. Another thing, the woman's capsule pressure should be low. Okay, the capsular pressure should be low. Okay, you can read capsular pressure should be less. So this is simple thing. If you want to push these particles in this, okay, if you want to push this particle into this. So first thing, it should be fit into these gaps. What I am drawing here, it have to be fit into this gap. Second thing, the pressure which you are applying in this side, this pressure should be higher. Okay, this pressure should be this pressure should be higher. That is a arterial pressure. And the pressure which is going from here, this is also applying one pressure now. This is also applying one pressure. From this side, this pressure, opposite pressure should be lower. Okay, that capsular pressure should be lower. Then only this solid particle can enter into this. Got it? This is the important thing. Now, if you will see, in complete kidney, if I am saying kidney, it will contain both kidney, right and left kidney. If complete kidney will you will see, this complete kidney can filtrate 125 ml per minute in each in all nephrons, okay, per minute. So in complete in 1 hour it will be how much? Around 7.5 liters. Okay, in 1 hour, and in complete day 180 liters. And we have only blood only we have only 5.4. So are you drinking anytime 180 liters? No. No, no. So how you can release this much of 180? And if anybody will release this much, they are not going to be survived. They will be definitely they will be die. Okay. If they will be die, the what is the purpose of making? What is the purpose of making of this 180 liter? Okay. So the purpose is that it should be passed everything into the process. Okay, it have to be passed everything into the process, into the filtration, and then only most of the things like 90% of the 90% of this filtrate will be reabsorbed. Okay, 90% of the filtrate will be reabsorbed. So only 90% will be reabsorbed. Okay, 90% will be reabsorbed, only 1.5% liter around will be go for the urination. Okay. Now, this will be the very important thing. What we are going to see now. Okay. So, as you people are not, you people are following. Anybody have any doubt? No doubts are there. So this is the very important thing which I am going to write, and I can draw also, but it will take very much too much of time. So you just write in proximal tubule. Okay, in proximal tubule, what will be reabsorbed? Okay, in proximal tubule, what will be reabsorbed? 70% things will be reabsorbed. Okay, and what will be reabsorbed? Like ions, then ions, glucose, glucose, and amino acid. If you are writing ions, don't write potassium. Okay, except potassium and plus. Okay. So these ions cannot be reabsorbed. What are the ions will be reabsorbed? Like chlorine, sodium, okay, bicarbonate. These ions will be reabsorbed. Got it? And secretion will be occur. What will be secreted? So these things will be go for the secretion. Secretion you can write urea and then potassium, hydrogen will go for the secretion. Okay. If I'm secretion, I am writing. It doesn't mean the blood will secrete anything. We are seeing in respective of the filtrate. So we are secreting in the filtrate. Okay, this and reabsorption from the filtrate. Then second is first is that proximal tubule. Then loop of Henle. Loop of Henle is divided into ascending and descending. Okay, loop of Henle. So this is loop of Henle. Okay. So first is that descending loop. Okay. So descending loop will only reabsorb. Descending loop will only reabsorb water. It will be permeable for the water. You just write like this also. It is permeable for the water, impermeable for the ions like sodium. Okay, so it will be this question is already asked, so I am telling you to write reabsorb like water, urea, and then descending ascending loop. This is the descending loop means like this. So this is your descending loop. There will be so much of diffusion. Reabsorption means taking out of the water. You're taking out this water. Okay, got it. And in ascending loop, what will write? Ascending loop. In ascending loop, you have to write the reabsorption will be occur of sodium. Okay, reabsorption will be occur of sodium, chloride, potassium, like other ions, like calcium, chloride. These all of the reabsorption can be occur, and secretion will be occur of secretion. Secretion will be of yes. Okay. So this hope you understood. Then DCT. DCT will be the third distal convoluted. Okay. DCT. In DCT, you will the DCT absorption will be reabsorption The reabsorption will be occur for the sodium and the chlorine and like these ions. So the sodium, chlorine, and secretion. Secretion usually it will be not occur here, but you can write express or potassium. Okay. So usually the S plus will be always secrete. So in DCT, usually secretion will be not occur. You can know this. In NCRT, it may written, you can write. Okay. DCT and then collecting that. Okay. Collecting that in collecting that reabsorption will be occur of the some drugs, some urea, collecting drug, it will reabsorb urea and water sometimes these are the and drugs. Okay. Some drugs will be reabsorbed in the collecting drug. So this is all about the urine formation. Now your urine is concentrated and we have to read something more about that osmolarity, how it will be balanced, why it is required to be reabsorbed these things, and that we will read in the counter current mechanism. Okay. Hope so you understood these things. If you have any doubt, you can ask me. If you want to continue this class, then also you can say I can continue. If you want to leave, then also you can say. So any doubt is there? No doubts are there. You have to just type, like type no, no. That's it. No, no doubts are there. Okay. So, shall we continue this class or tomorrow? Tomorrow we will read in the class. And thanks for being with me till now. And if you have any doubt, you can ask me. And those viewers, new viewers who are watching this video, you can take these live classes in U Biology app also, or you can attempt some questions which will be there in the U Biology from related to this topic. So, again, thanks for watching. We'll meet in the next class. Jai Hind.